So let's just take a look at the overview of this presentation and what we'll be covering. So the workshop is specifically about how gene editing technologies can save the lives of cystic fibrosis patients. So we are aware that we have students of different ages joining us. So the material will be more complex as we continue with the webinar. The first half of the workshop will be focusing on a simplified explanation of what gene therapy is, what is CF, and a case study that relates to the two topics. And then the second half of the webinar will be an interview with a high school student, Mitra, who is also the founder of startup nanotech and biotech companies. So he'll be discussing how students can really take initiative and find opportunities and become leaders in the healthcare industry similar to him. So we want our viewers to really feel empowered and play an important role as leaders in the future of healthcare and medicine. So just to introduce ourselves, Generation Science is a student-led nonprofit organization that aims to promote innovative thinking amongst molecular bio and med-sci enthusiasts. So let's have our speaker, Nasi, who will be diving into what is CF and really explaining the process of gene therapy and gene editing technologies.
All right. Cystic fibrosis is an inherited genetic disorder that affects the function of epithelial cells that produce mucus, sweat, and digestive juices. These liquids are supposed to be thin and slippery, but a defective gene in the body makes them thick and sticky. Because of this, these fluids can't act as lubricants in the body and instead clog up passageways, especially in the lungs and pancreas. Some symptoms of cystic fibrosis, or CF, are a persistent cough that makes thick mucus or sputum, wheezing, not being able to handle exercise, repeated lung infections, inflamed nasal passages or a stuffy nose, repeated sinusitis, poor weight gain and growth, or intestinal blockage, particularly in newborns. Cystic fibrosis, or CF, is also an autosomal recessive disease. This is just a fancy term saying that two copies of the recessive allele have to be present in order for the disease or trait to develop. In other words, if one of your parents has the cystic fibrosis gene and the other one doesn't, you won't inherit cystic fibrosis. It's also possible to have a copy of the cystic fibrosis gene without actually having the disease itself. This makes it possible for two parents without CF able to give their child CF because, although they don't have the disease, they have the gene in their body, which they might have inherited from their parents. Now for some numbers. Each time two cystic fibrosis gene carriers have a child, there is a 25% chance that the child will have CF disease, a 50% chance that the child will be a cystic fibrosis gene carrier but not have the disease, and a 25% chance that the child will not be a carrier and not have cystic fibrosis disease. As you can see, it's a bit of a coin toss in terms of whether you have cystic fibrosis or not. Unfortunately, cystic fibrosis is a progressive disorder, meaning that it only gets worse as time goes on. However, many CF patients live into their late 30s and 40s as screening methods and treatment options become more widely available and accessible. As I touched on earlier, cystic fibrosis is caused by a gene mutation. This mutation can be found in the CFTR gene, short for cystic fibrosis transmembrane conductance regulator gene. It lies on chromosome 7 and causes the production of proteins or channels responsible for regulating the movement of chloride ions in and out of the cells. CFTR protein function may be abnormal because of a lack of production of the protein channel itself, failure to reach its site of action due to misfolding of the protein, conductance, unusually low channel numbers, decreased half-life, or defects in gating. Now, to understand gating, we have to understand why it's called gating. The CFTR protein is typically shaped like a gate, which allows chloride to flow in and out of the channel when it opens and closes. Gating mutations lock the gate in the closed position so that the chloride can't pass through. Cystic fibrosis can be diagnosed through newborn screening in the first month of life. Otherwise, people may not know they have cystic fibrosis until they start experiencing symptoms. A doctor will order two tests, a sweat test and/or a genetic test. A sweat test will be ordered because cystic fibrosis patients actually have a higher volume of salt in their sweat compared to the average person. These tests will be used to confirm the presence of cystic fibrosis disease. According to the Cystic Fibrosis Foundation Patient Registry in the United States, more than 30,000 people are living with CF in America and over 70,000 live with it in the world. Around 1 in 2,500 to 3,500 Caucasian babies are diagnosed with CF in America. The ratio among African Americans is at 1 in 17,000 births, and for Asian Americans at 1 in 31,000, which is interesting to see. As you can see, it's an uncommon condition. However, it's important to be able to help those that do have the disease, and this is where gene therapy comes in. Now, let's do a 2-minute quiz to see if you guys understand the symptoms of cystic fibrosis. We have 3 people here. Person A, a male 16-year-old who informs the doctor he keeps vomiting. He has been experiencing a lot of stomach pain since yesterday. Person B is a 45-year-old male who reports a lot of back and knee pain. He states that he has been having a lot of muscle pain that won't go away. Finally, person 3, a 21-year-old female who reports salt-tasting skin and a stuffy nose. She reports feeling fatigue, having difficulty exercising, and has lost a lot of weight in the past months. Which one of these is likely to have cystic fibrosis?
Everyone got it correct, so great job, guys.
Good job. Alright, introduction to gene therapy. Gene therapy is a new and unexplored method of treating cystic fibrosis. What gene therapy does is that it supplies genes to perform functions your body can't otherwise carry out, such as making helpful proteins to treat a disease like CF. This new gene is also blocking other genes from producing unwanted products. Essentially, what you're doing is inserting a bad gene, or a good gene rather, and kicking out the old bad one. In the case of cystic fibrosis, you're transferring copies or clones of a healthy CFTR gene to the epithelial cells in the airway, and therefore treating the patient. It's important to note that the good gene will not be passed down to any children the patient decides to have. Gene therapy is a treatment for the individual suffering from CF and will help the body fight the disease, but it will not prevent the mutated gene from being passed on among generations, unfortunately. Gene therapy is usually used when there aren't any other viable cures for the disease, because there are some risks associated with it. The healthy gene can be either inhaled or inserted intravenously, meaning it's inserted through the bloodstream. The process of getting the healthy cell into the pulmonary cells is difficult, since the cells are covered by a protective membrane. And what's worse is that it's even worse with cystic fibrosis patients because they actually have a thick, heavy mucus layer around their cells. Once the cell, or the gene rather, is in the body, the gene is directed into the lungs through vectors. Vectors are DNA molecules that are used as vehicles to transport the healthy cell into the lung. CRISPR is a technology that allows scientists to perform gene editing. It locates defective genes and modifies them so that they are able to perform their designated function. It can also turn off genes without modifying their sequence. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. The name is derived from the fact that this technology adds the viral DNA to its own genome in order to guide the Cas proteins to find the mutated site. Here is a gene therapy step-by-step process using CRISPR. 1. The guide RNA, or ribonucleic acid, seeks out and attaches to the Cas9 nuclease. The guide RNA will act as a tool for comparison for the Cas9, as the Cas9 will replicate its structure to act as the good gene. 2. Once it's done this, the Cas9 release can act as a detective to seek out and bind to the defective parts of a genetic sequence. Once the target site is reached, the Cas9 nucleus will unwind a certain section of DNA. It will compare the guide RNA sequence to test for a match. If a mismatch is detected, the Cas9 has found a site of mutation. 3. The Cas9 nucleus will cut the site of interest. And finally, the repair template that contains the healthy gene sequence is inserted. And that's it. Other than cystic fibrosis, gene therapy has also been used to treat cancer, heart disease, diabetes, hemophilia, and AIDS. It's a new and promising method for treating conditions that many people have believed were incurable. Now, we'll be creating breakout rooms to discuss your thoughts on CRISPR and other gene editing technologies. So the question is, do you think gene editing is morally correct? What pros and cons are there to the topic? And we'll take up your answers in 5 minutes.
Nancy, I think you can go ahead and share the answers.
Sure thing. Actually, I'll go through Arjun's comment. He just commented, almost any DNA sequence that is mapped or understood well enough to be created in vitro and be delivered properly is able to be modified in theory.
Yeah, that's a great point.
For sure.
Mm-hmm.
Yeah, so whenever gene therapy is discussed, the issue of ethics is bound to pop up. I mean, myself, when I was first introduced to gene editing a few weeks ago, I was talking to my friend who's involved in CRISPR research, and my first question was, do you think it's right to be able to edit genes? And the answer to that question, and many of these questions, is that we don't really know, right? Like, it depends on every person and their values and ethics, so you just have to weigh the pros and cons of gene editing with your own personal values, for sure. Some other questions that pop up when we talk about ethics is, what if the wrong gene is edited, which could cause potentially devastating effects? For example, if it mutates a perfectly healthy gene, it can lead to a wide array of serious health complications. Next, how safe is the gene editing procedure? As it's a relatively unexplored field, side effects are still being discovered and studied. How will the healthcare system allow for equal access for this type of therapy? Because this might be expensive and only wealthy patients may be able to access it. In the event of a security breach, how will privacy be protected? And I know in my breakout room this was discussed as well. And finally, what is the property status of information derived from gene editing? Which is an interesting point too. These are only a few out of many concerns that have emerged with the growth of the field of genetics and gene editing and are definitely interesting to look at and consider.
Now let's take a look at a real-world example of how Crystal Biotech is using gene editing technologies to revolutionize healthcare. The study was aimed to find a way to treat cystic fibrosis through gene therapy by delivering a non-mutated version of the gene to patients with CF. Crystal Biotech hypothesized that the introduction of the KB47 gene would be able to induce the expression of normal CFTR protein in cell models. This gene was delivered through vectors. Vectors are basically DNA molecules that act as vehicles for genes because they can carry and transport the healthy gene to the desired location. In this case, a modified version of the herpes simplex virus 1 acted as the vector to carry the gene. So as mentioned before, the platform contains an engineered HSV1 vector, also known as the herpes simplex virus 1. This modified HSV1 is a replication-defective viral vector that can efficiently penetrate a broad range of skin cells. This means that it is able to deposit the healthy gene into cells, for example, a full-length CFTR gene, but it will not replicate and infect other cells. The HSV1 is also a non-integrating viral vector. There are two types of viral vectors, non-integrating and integrating. The difference is that integrating vectors have their DNA/RNA permanently fused into desired chromosomes. This basically means that the DNA/vector that is already incorporated into the chromosomes will stay inside both daughter cells during mitosis. Just like the name suggests, non-integrating vectors' DNA is never incorporated into the chromosomes. This means that when the cells undergo mitosis, the vector DNA will not be able to stay inside the daughter cells. The main difference between the two is that integrating vectors will stay inside during mitosis, while non-integrating vectors will not be passed down to any of the daughter cells. CRISPR Biotech developed a gene therapy platform, the Skin Targeted Delivery Platform, or STAR-D. It consists of an engineered HSV1 vector and skin-optimized gene transfer technology to form a therapeutic vector. It introduces one or more therapeutic genes into the patient, in this case, the KB407 gene. So now I'll go over the process of the gene therapy that was used in the study. So first, the STAR-D platform targets the mutagenesis of HSV1, ensuring a platform that is both harmless and effective for the delivery of effector or effectors into human skin. Next, a therapeutic vector modifies the STAR-D platform through the insertion of one or more therapeutic effectors of interest. Then it is formulated into a topical gel or intradermal solution, also known as the drug product. Finally, it can be applied locally to the skin for treatment, using a gel, which the patients can apply directly onto the skin, or a needle that can be inserted into the patient's body. An advantage of this approach is that it can work equally well regardless of a person's particular CFTR mutation. Some targeted CF therapies only work with specific mutations, and more than 1,700 CFTR mutations are known in this disease. All the new data mentioned on KB407 came from in vitro experiments, performed on cells in a laboratory setting. In vitro is Latin for in the glass. In one experiment, researchers found that in lung cells that were lacking CFTR, KB407 treatment successfully restored protein production as assessed by multiple molecular tests. Another experiment conducted using organoids. Organoids are cells grown in culture using a particular three-dimensional structure that more closely resembles how cells are organized within human tissues. Scientists have found that organoids are able to model the retina, brain, intestine, and other organs of the gastrointestinal tract, liver, kidney, among several other organs. Organoids were inserted to mimic healthy gene expression in CF patients. They also make it easier to control and manipulate the gene during the experiment. The results of these trials demonstrated that the KB407 stimulated the synthesis of the CFTR protein, despite being in relatively low doses. The CRISPR Biotech researchers wrote that, taken together, these data indicate that KB407 capably infects relevant airway epithelia, effectively produces functional human CFTR, and molecularly corrects multiple CFTR defects, supporting application of KB407 as a novel, broadly applicable gene therapy for the treatment of CF. In a press release, Krish S. Krishnan, the company's chairman and CEO, said that we are encouraged by the preclinical data to date on KB407 with potential to treat cystic fibrosis that demonstrates the breadth of our STAR-D platform, and are excited that we have an opportunity to share this. In conclusion, this study/experiment had made it a possibility to allow cystic fibrosis to be treated with gene therapy. CRISPR Biotech also expresses the continuation of preclinical studies of KB407 and plans to try requesting trials in people starting in 2021. KB407 has great potential to become a safe and effective way to treat patients with cystic fibrosis, especially if technology continues to develop at this rapid rate. There could be further advancements made to KB407 that could improve the patient's experience with CF treatment. This study demonstrates how quickly technology and healthcare are evolving around us. We should all be grateful for the time and effort that is put into improving technology and healthcare. Apart from STAR-D's gene therapy for CF, many treatments are now available thanks to new technology. For example, laser eye surgery was invented in the late 1900s, but there are still improvements being made to the surgery that allows the surgeon to have higher precision. STAR-D's developments alone could impact over 70,000 people who live with CF in the world. So this data reminds us of how essential technology is to revolutionizing healthcare.
Thank you, Elaine and Nancy, for educating us about the applications of gene therapy and gene editing technologies. Next, we'll be interviewing Neil Mitra, the CEO and founder of Mitra Biotechnologies and Icane. During this interview, we will be discussing the future of biotechnology and healthcare and how students can seize opportunities on their own and take initiative to become future healthcare leaders. So, let's start with the first question. First of all, hi Neil, how are you doing today?
Doing well, thank you.
Nice. So, could you elaborate on the goals of both organizations and overall what each of your things is about? How is your team revolutionizing healthcare?
Sure, absolutely. So Icane essentially produces socially assisted robotics for the visually impaired, and Mitra Biotechnologies is aiming to create rapid point-of-care diagnostics for individuals with heart disease. And essentially our team is quite diverse, with quite a diverse range of qualifications, and we have a strong scientific advisory board for both of these companies, and we plan to take our products to the market soon.
That's really interesting. Could you explain specifically how your technologies help patients? What processes or techniques are involved?
Absolutely. So for Icane, we are currently in our beta testing platform. We just got seed funded a couple of weeks ago. So now we're getting our beta test out with our current prototype and getting LOIs, so license of intents, from these consumers. For Mitra Biotechnologies, essentially what we do is we're reducing the amount of time taken to do blood plasma separation. So we're currently the only and the first device on the market to support cardiovascular disease patients with rapid point of care. The way we're able to do this is minimizing the time for plasma and blood separation. So conventionally, for separating these plasma and the blood, you would have to use something known as a centrifuge. And a centrifuge is quite expensive, and it takes around 15 hours to separate the two. The reason why it's important is because in the plasma itself, you have these biomolecules that you're trying to detect for your tests, regardless of whatever blood test you do. So what we are doing is we're reducing the time span of that 15 hours to just 10 seconds, and we're doing it a completely paper-based platform. So it only costs about $2 or $3 to produce.
Wait, so this wasn't part of the formal outline, but spinning off of that, what exactly is the methodology for you doing this?
Yeah, so for Mitra Biotechnologies, we're using a nanomaterial-based paper. So we're integrating a whole bunch of nanomaterials and doing a lot of fabrication techniques to create a paper that is essentially able to separate the two.
Okay, cool. Yeah, I saw something similar. It was like UC student created super cheap paper centrifuge that costs 5 cents. But why even have a centrifuge when you can create a paper that can do the same thing?
Yeah.
So what would you consider the greatest achievement of your organization?
So for Icane, so far it has definitely been in terms of getting our seed fund round ready and well as getting our beta test ready. So that was one of our two most significant accomplishments. For Mitra Biotechnologies, currently we are seeking investor money to get us into the MVP stages, as well as to get us through to our initial customers and navigating regulation. So by that standpoint, I would say Mitra Biotechnologies is doing pretty well. We have a successful proof of concept, which is in vitro at the current moment, and we hope to bring it into the market in the next couple of years.
Yeah, to answer one of the questions in the chat, are the results congruous with what's achieved by using a centrifuge?
So could you elaborate a bit more in terms of that question?
For Mitra Biotechnologies filter that's paper-based rather than using a centrifuge, are the results for detecting those plasma particles the same as
Oh yeah, definitely.
what a centrifuge would have?
They're way more selective in terms of getting the detection, since we're using nanomaterials and we're using quite rapid nanomaterials. Nothing is more effective than nanomaterial filtering. It is the most effective point of filtering that you can get. It's to the nano level size. So it's quite easy to filter using our system, and it's far more selective than using a centrifuge.
What fostered your passion for both organizations?
So for Mitra Biotechnologies, I would say it's mostly because since I'm a member of the South Asian community, one of the leading causes of death for the South Asian community is heart disease. And I looked into it a bit more, and I realized that overall in the entire world, around 18 million people die every year, and they don't have any system of monitoring their levels. For example, with diabetes patients, they have glucometers. They can monitor what their blood sugar levels are, and they can create a plan according to do so. However, for heart disease patients, there's none. So I decided to create the first one. And then for Icane, essentially my own grandmother is visually impaired, and there was nothing there to actually help her to navigate effectively. Current solutions only allowed her to see or to understand obstructions around her, but they couldn't actually navigate hers. So what I decided to do was create a robotic guide dog, and that's what Icane does.
So how did you approach researching and developing both of these products, and do you have any recommendations for students on how to access those resources?
Yeah, sure. So for Icane, it was mostly a completely self-funded project. Since that was mostly hardware-based, it was mostly learning like hardware technologies, how to code in C++, getting Arduinos, Raspberry Pis, getting all these parts and hacking it all together. Then for Mitra Biotechnologies, since this is a much more advanced topic, what I had to do was get in contact with a couple of professors. Here at the University of Waterloo, I got in touch with 2 professors in the area of chemical engineering and in biology, and I worked in their labs. So I definitely encourage students to go out and reach out to professors at your local nearby university, asking them if, whatever you're trying to do, if you could like research in their lab. And most professors would tend to look at it, but not respond. And then eventually, after a bit of trial and error, you'll eventually get professors who would be interested in your project. And yeah, that's probably like the best advice I could give.
So, who's helped you get where you are, and how can we replicate that?
Mm-hmm. So, in terms of helping me to get where I am, I would say it's mostly like a large part of it was getting the opportunity. So, just asking people, like having the ability to ask individuals and asking kindly and telling about yourself in a presentative way can go a long way in terms of getting these opportunities. So the biggest opportunity for me, especially for Mitra Biotechnologies, was getting lab access and working in the lab of these very high-level university labs, which was quite the experience, as well as I got quite a load of connections from that, especially in the whole entire university network. So definitely the aspect of networking is a huge skill to have, especially in today's world. It is arguably the most important skill to have, which is talking to people and being able to present yourself, essentially.
I definitely agree with that. That networking, I would just by myself, I'd say networking is one of the most important things you can do to get started in any venture, period. So what do you think some major issues in today's healthcare system are? So
the most out of the bat issue is if you look at the current companies, say, a bot or Roche Diagnostics. I'll use diagnostics as a general viewpoint, is that their diagnostics are simply not very capable in a scenario where it comes to user-focused use. So an average individual, they cannot use these high-level diagnostic equipment, especially for heart disease patients. The only time they can ever have a point-of-care tool is after they get a heart attack, and they have to be rushed into the hospital, and that is when they use these point-of-care devices. But at that point, it's effectively worthless. A person cannot know before when they're having a heart attack. And there's like a whole bunch of major flaws. Like these devices cost over $1,000. They're effectively worthless. And there are many examples of this in the entire healthcare industry. You have these so-called technologies which are meant to help people, but in principle they cannot. And yet that is still generating revenue for these companies. So that is one key area that requires disruption in the healthcare technology sector.
So what do you think the future of biotech will look like? Will there be more startups like you're doing?
I would say startups would have a huge role in terms of the biotech industry. Like the biotechnology industry is such a recent industry comparatively to others. And we're sort of at that stage that we don't really know yet, because it's such a... Biotechnology is at the stage at which computing was, say, about 40 or 50 years ago. It is so new and recent, and seeking into the future, it's quite not feasible at this current moment. But I think gene editing, as you guys have said previously, would definitely be a key role as to providing the future of biotech.
So do you think that in practice, in the clinical setting, the healthcare industry will have a shift towards technology and engineering in the future?
Absolutely. I mean, the biggest shift will definitely occur when it comes to artificial intelligence. Consider your average physician. Your average physician makes a diagnosis based on how many cases they have already previewed and based on their medical knowledge. And artificial intelligence can store hundreds of thousands of patient case data and be able to learn much more quickly than than your average individual. So definitely technology would play a really key role in terms of getting access to healthcare for all. Yeah.
Yeah, definitely. Adding on to that, like, just recently I started a fellowship with this company that does medical imaging using artificial intelligence. And the just extreme power that artificial intelligence has can go even beyond like a radiologist or multiple radiologists. And diagnosis based off of artificial intelligence in the future will most likely become even more accurate than the human.
Yeah, definitely.
So what role do you think biotech plays in the current medical world? What impact do you believe being in biotech has?
I would say the biggest role is definitely helping people. Like, biotech has the scalability factor of a regular technology company at the same time as the core principles of science. And combining those two with that scalable factor, you can create a lot of change, especially for providing to those who cannot normally afford healthcare is one of the biggest areas. For example, like the high growth economies that are soon to happen, for instance, in India, as well as like all these developing countries in Africa. It is quite important to keep in mind that healthcare there is still a bit fragile. So biotech can definitely play a huge role in terms of these suburban communities. And we'll definitely see that shift maybe 10 or 20 years from now.
So I heard you saying about how heart disease is somewhat neglected, and heart patients can't typically have ease of access to tests and things. So besides that, what's an area of research you think we ought to pay more attention to, and why and how should we approach it?
Biggest sector of research we should primarily focus on is definitely like prevention of the entire disease itself. So how can we create a system that prevents all of these diseases? And one of the key pointing tools that we have is looking at just basic nutrition. So food, our diet, the way that we keep healthy is a key factor in terms of these diseases. And it's quite important to look into that factor. So preventing these diseases really starts from the basics, how well your diet is, how well your exercise is, and these core value principles in terms of physical health. And that's something that we have to look into quite deeply, as well as, avoiding viral infections. That's also a huge part. Viral infections kills a lot of people, especially in the developing world. And it's important to figure out how we can tackle these problems at a large scale and rapidly at a large scale.
So, of course, there's no shortage of things that we can be working on in the field. So what do you think is the biggest obstacle preventing people from taking initiative and doing these sorts of things? What are some ways we can overcome that, or what are ways we can overcome those obstacles?
The biggest is capital resources. Biotechnology is one of the most capital-intensive industries in the entire world. You require a lot of capital and a lot of funding in order to simply get a product into the hands of consumers. There's a lot of ethical processes, there's a lot of regulation in this industry, and it's important to keep that in mind. For example, if you take a general software-based idea, it's easy to scale, it's easy to create the actual product. Scaling is a bit harder. In the biotech industry, it's sort of inversed. To create the product, you do not know if the product is actually feasible, and at the same time, it has quite a lot of capital-intensive aspects to it. However, marketing the actual product and scaling it is quite easy in comparison to general software products. So that's a huge difficulty in the biotech industry, is providing that capital and that access to these individuals. And one of the easiest way that youth can access such capital and places to work at, as mentioned previously, is working in labs, working at universities, getting that experience right off the bat, so that in the future they could start their own startup, for sure.
So what do you wish you knew before you started your own businesses, and what's the most important thing to consider when starting a startup?
I would say definitely focus on the customer. Focusing on the customer is the primary goal of any business, and it should when you're starting out a company, your key idea should not be to raise capital or to raise money or to get a high valuation. it should be to really provide for the customer's needs. And if you don't provide for the customer's needs from the very start, you'll never raise money, or you will never raise a high valuation. So that's something to keep in mind. And at the same time, developing is such a huge part. So developing, getting customer feedback, iterating the product, and refining it is a huge part in terms of the initial process. Another thing that you have to look into is simply leadership. You have to lead an entire team, and there's a lot of project management principle which goes in to a very early stage startup. So leadership skills is something that is quite integral in terms of keeping your company's morale high, and at the same time developing these great products. So yeah, that's primarily what I wish I knew.
So what do you believe is the most important factor in creating any new product?
The most
Or
important,
startup?
yeah. So the most important thing is seeing if there's a customer demand. Like there has to be customer demand, or else simply nobody will buy what you're creating. So market research is the most essential part to any startup. So understanding to see if your consumers actually want the product that you're building.
So what sort of qualities would you think would help a person do such a thing, or create such a startup?
I would say you have to have a hunger and a passion. So whatever your passion is, you have to be really hungry for it, and you have to strive for it. And the world of business is quite cutthroat, and I don't put that lightly. It's a very competitive area, and it is stressful, and it is quite sometimes very hard to manage, especially if you have another commitment, for example, school. And you have to be very passionate about what you do, not just because of getting the money at the end. It's really about getting the journey to that point. So you have to be really passionate about what you do. You have to love what you do. Otherwise, you simply will not be able to do it, because it gets way too strenuous as you move along.
So what type of work environment does being in biotech provide?
Mm-hmm. I would say so like compared to tech industries, like I would say tech industries, if you look at Google's offices and all of that, it's quite like a playful place, for a lack of better words. And I would say for biotech companies, you see a whole bunch of people in lab suits working in the lab. And it's quite an interesting comparison to see those two industries. Like if you like being more centered in a way to the area of creating stuff that you want to create and getting that, finding a team of people that take you to market, I would say that biotech is definitely a very interesting place for you to look at. On the other hand, software is far more collaborative. People are easily able to pick up these skills. However, for biotech, you need to be really, really specialized in certain things in order to excel in that industry. So if you like specializing in a certain area, you like taking the time to learn more about certain principles, biotech would definitely be a first option in terms of the workplace environment.
So what role did clinical research play in the development of one of your products?
Oh yeah. So for sure in IVD development, which is in vitro diagnostics, at least for Mitra Biotechnologies, there's a lot of testing which has to go through to see if the diagnostic works, and a lot of FDA regulation. And those are quite capital intensive. You have to pay the volunteers who are going to use your device. You're going to have to pay the FDA to get an application, and you have to pay all these people, all these legal and administrative staff to create these applications and these processes. So it's definitely a very capital intensive process in that retrospect. And in terms of clinical phasing, there has to be a lot of work done. You simply cannot have a diagnostic which fails in principle when it goes into the hands of consumers. So that's why in order to create that trust with the consumer, you have to have a high level of research testing. And the biotech industry is quite brutally known for their standards of clinical procedures.
So where do you imagine yourself with these research projects in the next 10 years?
Mm-hmm. So I don't really like envision myself in it. Like I don't really envision things within the next 10 years or so. I like to take it in small periods. So for example, like in a year or so, I want to see Icane get into the phases that we can start actually producing our products in the mass scale. formation of Biotechnologies, we could probably see a completely successful MVP prototype, which is about to go into the phases of FDA regulation, as well as getting into the hands of initial beta consumers. So that's what I see in the next year or so for both of these startup companies that I'm doing. In the next 10 years, for that, I guess we'll just have to see.
Adding on to that, when you say you want an MVP, you mean minimal viable product?
Yes,
for
yes,
yeah.
yes.
So how is it that in the biotech field, how does the notion of a minimum viable product work, especially when your goal is so specific?
Mm-hmm. Yeah, so for a minimal viable product in the biotech industry, it has to be something that a person may be able to use right at that point. So a proof of concept is something that is probably done using a sort of like a simulated blood material or something. When it goes to an MVP stage, it is something that is a device that you can see, and a person, if they wanted to, they can prick their blood and like use it. But for proof of concept, it is quite similar to an MVP. However, you're not using actual blood, you're using some sort of variants. At the same time, you might have ex situ procedures. So a lot of these chemical procedures can be separated from the actual device itself to master that principle for the chemical detection and then combine it later on. So to create the MVP stages, it's not that capital intensive, first of all. So it's probably one of the least capital intensive processes, going from a proof of concept to an MVP stage, because it's so similar. And after that, the next huge stage is getting that into the hands of consumers, which would be through a lot of regulation and beta testing, which is very capital intensive.
So how do you establish credibility for your organizations? And how do you get the word out and build that trust that's needed?
Yeah, so you have to network. Again, you have to get you have to know in and out of what you're doing. That's the first thing. You have to know in and out of the technology. You have to know in and out of how basic biology works. And then you go to like a whole bunch of professors and you pitch what your idea is. Another thing that you have to keep in mind is to make sure your IP gets protected. So always file a patent, especially if it's hardware based. And that way it's protected. And then from there, what you're going to do is you reach out to people, you network, you find people in the healthcare industry who would be willing to act as mentors and advisors. And then you establish an advisory board of these strong people, and that gives you credibility. And it's really that simple, but it's hard to do.
So how do you get the money to get these patents?
So luckily, like for youth, it's quite easy. You're pretty much in the section known as a micro entity status. So you pretty much have to pay like 1/10 of the cost of a patent that you would normally have to do, since you do not have a taxable income. And it's quite easy to pay those off. Like they usually range between $100 to $200, depending on what sort of patent you're applying for.
So what are the biggest challenges you encounter with your startups, and how do you overcome and adapt to those?
The biggest challenge easily is managing a team, because you're going to have so many people that join it at first just for the sake of putting it on their resume or something, especially at a high school stage. Most people, they just want to do it for the sake of getting into a university or something like that. And simply, in order for a startup to succeed, you cannot have the intention of just doing it for the sake of university. You have to have the intention of actually getting this product to market. So you're going to have a lot of people who join your startup and leave your startup. And you have to be quite protective of yourself as the primary founder. You have to understand how equity and share distribution works. You have to understand how stock options work. And a lot of business fundamentals you have to understand for the beginning. And that's a key important aspect to keep in mind. And that was definitely one of the biggest difficulties for me, primarily because I come more or less from a science and engineering background, not so much in like a business background. So I had to pick up and learn those things quite quickly.
So how exactly do you think that students should get their network started? Because it's like that. It's like the paradox with getting your first job. Like jobs require experience, and experience requires a job. So how do you start this networking and like getting your first opportunities?
So the easiest way to do it is start building stuff. Like start building projects. Create like fancy name for your projects. and put those on a LinkedIn. Like the easiest way to network now is getting LinkedIn, get in contact with CEOs, get in contact with professors that you want to be with and research with. And like I would say, strongly recommend to have like an email address, a formal email address that you contacted all these people with. And I wouldn't like, that is the number one thing that I'd recommend. Probably like sending out cold emails to like get to know other people well. And that's how you start to begin to network, at least during the COVID-19 pandemic. If it's not the COVID-19 pandemic, you can definitely enter and register for like all these huge tech conferences that happens, and all these other conferences related to science that any individual can sign up on. Probably like a small admission fee, but that's about it. And network with everybody there.
So how do you handle the stress and the feelings of being overwhelmed? How do you manage both organizations and school?
Yeah, so I would say like you have to love it. You have to love the constant pressure. You have to love like the constant rush that you get from working at companies. At the same time, you also have to somewhat like school. So you have to realize that also school is a bit important in terms of, you know, getting into the university you want to get into. At the same time, like managing all these other extracurriculars and companies, especially that you want to take and with you to these universities, because the biggest thing in universities is getting the access to the network that universities have. And that university network combined with the startups can really accelerate that forward.
Well, thanks, Neil. I think that's all the time we have for today, so I'll hand it over to Shay.
Thank you, Neil, for joining us today. You really provided some really helpful advice, and I hope that students can really learn from the session. Make sure that you guys follow their team on
Thanks for having me.
Yeah, make sure you guys follow their team on Instagram and LinkedIn. Their Instagram handle is Mitra Biotech on Instagram, and you can also search them up on LinkedIn by searching up Mitra Biotechnologies. So I hope everyone had like a great time learning about topics that they're passionate about, or even they're able to learn something new that they never knew before. And that just brings us to the end of our presentation. So if you guys have any questions, please email us at contactgensci@gmail.com. And be sure to follow us on Instagram and LinkedIn if you guys haven't already.